Bacterial pathogens aggressively multiply and adapt to their environments. To survive this constant threat, host organisms evolved a robust, immediate chemical defense system. The biological standard for this defense relies on antimicrobial peptides, or AMPs. These small, positively charged molecules are deployed by the innate immune system to neutralize bacterial invaders on contact. While highly effective in nature, natural AMPs contain a critical structural flaw that prevents them from becoming viable commercial therapeutics. They are highly susceptible to rapid proteolysis, meaning enzymes in the body quickly degrade them before they can clear a systemic infection. To bypass this biological fragility, researchers at Brigham Young University sought to synthesize a chemical alternative. Their work resulted in the creation of ceragenins, also known as cationic steroid antibiotics, or CSAs. Ceragenins are engineered by taking a rigid, proteolysis-resistant sterile backbone and attaching cationic amino acids to it. This specific charge distribution creates a facially amphiphilic morphology. The molecule possesses distinct hydrophilic and hydrophobic faces, mimicking the exact shape and charge profile of natural AMPs. By utilizing a sterile backbone instead of a peptide chain, ceragenins replicate the lethal efficacy of the innate immune system while replacing its biological fragility with synthetic durability. The prevailing assumption in microbiology is that amphiphilic agents must physically permeabilize the bacterial membrane to achieve lethality. In the standard mechanical model, the positively charged agent binds to the cell wall, inserts itself, punches a physical hole, resulting in catastrophic solute leakage. Ceragenins are highly lethal to bacteria, but experiments show they do not actually cause this inner membrane permeabilization. Microbiology researchers at Vanderbilt University Medical Center flagged these exact anomalies, observing rapid cell death without the expected solute leakage. Resolving this paradox requires shifting the investigation away from physical holes in the membrane and looking closely at underlying biophysical disruptions. The true mechanisms of ceragenin lethality rely on two specific processes: cytoplasmic membrane depolarization and anionic lipid clustering. Bactericidal lethality does not require physical perforation. Proving this requires a shift in how we measure and understand synthetic antibiotic efficacy. To test the permeabilization theory directly, researchers utilize a genetically engineered strain of bacteria known as E. coli ML35P. The cellular architecture of ML35P features a distinct outer membrane, a middle periplasmic space, an inner membrane, and the internal cytoplasm. This specific strain is engineered to house two key enzymes in separate compartments: beta-lactamase in the periplasm and beta-galactosidase in the cytoplasm. The assay uses a chromogenic reporter called nitrosophon. Because nitrosophon cannot normally pass the outer membrane, its cleavage by beta-lactamase acts as a strict indicator that the outer membrane has been breached. A second reporter, ONPG, is used to test the inner membrane. Cleavage of ONPG by beta-galactosidase proves that the inner membrane has been physically compromised. This graph plots time against absorbance to track permeation. Exposed to ceragenins, we see a rapid spike in nitrosophon cleavage, confirming outer membrane permeability. However, the inner membrane test tells a different story. The ONPG absorbance line completely flatlines over 30 minutes. This flatline persists even when the concentration of the ceragenin is pushed well above the minimum bactericidal concentration. Despite this inner physical barrier remaining entirely intact, the bacterial cells still undergo rapid death. The empirical data from the ML35P assay proves ceragenins bypass the need for inner membrane permeabilization. The traditional pore formation model is incomplete. If large molecules cannot pass through the inner membrane, the cause of death must stem from an electrical failure rather than a physical one. Researchers measure the electrical potential of the cytoplasmic membrane using a specialized cyanine dye called dye SC25. When incorporated into a healthy polarized membrane, this dye loses its fluorescence. If the membrane loses its polarization, the dye immediately glows highly fluorescent. This graph tracks fluorescence intensity over time. When the dye is applied and the ceragenin reaches its minimum bactericidal concentration, we observe a sudden spike in fluorescence. This signifies a total collapse of the membrane's electrical potential, reaching up to 80 to 90% depolarization. The inner membrane maintains its physical integrity against molecules, but it completely loses its ability to hold an electrical charge. Ceragenins execute their targets by causing a lethal electrical failure. They short-circuit the bacterium without punching a physical hole. We must determine what physically causes this electrical failure if no pores are formed in the lipid bilayer. The critical variable that determines a bacterium's susceptibility to this electrical failure is its specific membrane lipid composition. This chart compares depolarization across bacterial strains. Gram-positive bacteria with high PE concentrations are highly susceptible compared to heavily anionic strains like S. aureus. Mixed Zwitter-anionic and anionic membranes suffer up to 90% depolarization. This makeup presents a distinct biophysical vulnerability. To observe the lipid behavior under ceragenin attack, differential scanning calorimetry assays are performed on artificial mixed membranes. These tests revealed the mechanism of lateral segregation. The polychionic ceragenins electrostatically drag the negatively charged lipids away from the Zwitter-ionic ones. This forced clustering physically reorganizes the entire membrane surface, creating discrete, rigid phase boundaries between the separated lipid groups. This scatter plot illustrates the analytical data. We see a direct linear correlation between a molecule's net positive charge on the x-axis and the severity of the lipid phase shift on the y-axis. A ceragenin's lethality is governed strictly by its net positive charge, which dictates its ability to violently reorganize the target's lipid architecture. Uniting the data from these assays reveals the complete step-by-step mechanism of a ceragenin attack. In the initial phase, the facially amphiphilic ceragenin traverses the outer bacterial barrier and binds firmly to the cytoplasmic membrane. Because of the peptide's high cationic charge, this binding immediately initiates lateral anionic lipid clustering, drawing negatively charged lipids together. The newly formed phase boundaries between the clustered and unclustered lipids act as microscopic physical fault lines within the bilayer. These fault lines allow a fatal proton flux. Ions leak rapidly through the boundaries, entirely dissipating the cell's voltage gradient. Throughout this entire lethal process, the membrane remains completely intact and impermeable to larger polar molecules. When engineering synthetic peptides, the pharmaceutical industry must shift its primary metric of success away from physical permeabilization. True antimicrobial potency is driven by the capacity to induce lipid clustering and rapid membrane depolarization. This biophysical mechanism provides the blueprint for the next generation of synthetic antibiotics.